Hi guys. So today for my non-majors, we're talking about chapter 17, which is the immune system and disease. And we're also going to cover viruses within that. For my majors, it is actually two chapters. It is chapter 21 over viruses and chapter 42 over the immune system. So first off, viruses are non-cellular. And one of the requirements in order for something to be considered as living means they are made up of cells. So since viruses are not made up of cells, they are not living. They are not classified in any domain or kingdom, so they just do not meet the requirements in order to be living. There is still some scientific debate out there on whether or not they should be considered living because they do reproduce. However, they only reproduce when they are inside of a host. So the majority of scientists have agreed that they are not living. They're actually smaller than bacteria. They're about 0.2 to 2 micrometers in size. Micrometer is a millionth of a meter, so you could imagine how tiny that is. We have looked at bacteria in the lab, and you have seen how small bacteria are, and viruses are actually smaller than that. They are diverse in structure, but all viruses have two things in common, and you guys need to know this for your test, so pay attention. The two parts are the outer protein capsid and then the inner nucleic acid core. Now that inner nucleic acid core can either be DNA or RNA, which remember DNA is deoxyribonucleic acid and RNA is just ribonucleic acid. Now some viruses that infect animals actually may have an envelope covering that outer protein capsid. However, the two distinct parts that all viruses have, no matter what kind of virus they are that outer protein capsid and the inner nucleic acid core. They can be categorized by a variety of ways. Some of them are categorized by the type of nucleic acid they have, so whether they are DNA viruses or RNA viruses. Remember, RNA is single-stranded and DNA is double-stranded. They can be categorized by their size and their shape, or whether or not they have that outer envelope that I was just talking about on the ones that infect animals. So here's some pictures of some viruses. A shows you the tobacco mosaic virus, which infects tobacco plants. B is the adenovirus, C is the influenza virus, and D is the bacteriophage. Now, bacteriophages are actually pretty important because bacteriophages are actually viruses that infect bacteria. And bacteriophages can be used in genetic engineering. So I always remember it because it looks like a little alien to me for some reason. I could picture like a little alien machine walking around. So that's how I remember what a bacteriophage looks like. So bacterial viruses, again, bacteriophages. So these are viruses that infect bacteria. Then we have plant viruses that are only going to infect plants. And then we have animal viruses that are only going to infect animals. And remember, they may or may not have an envelope. So the structure of a bacteriophage there, it shows you the capsid head. And inside that capsid head is DNA. And then you have the tail sheath, the base plate, which is basically what they use to attach to the bacteria with their pins down there. And then their tail fibers kind of stabilize them, because what they end up doing is injecting their DNA into the bacteria. So they kind of have to be stabilized to do that. Again, they're not classified in any domain or kingdom, so we're not using the Linnaean system here. They do have the capability to mutate and adapt and evolve, and they can replicate. But again, they can only do these things while they're inside of the host. So that is why they are not considered living. They're considered obligate intracellular parasites. So obligate means that they have to do it. Intracellular means they're inside of the cell. And parasites mean that they feed off the cell, basically. So what they do is they actually take over the host cell's machinery for replication, and they have the host cells then replicate viral cells in a nutshell. And remember, obligate intracellular parasites, because you have to know that for your test as well. Now, how they replicate, there are two different ways. There are the lytic cycle and the lysogenic cycle. The lytic cycle is shorter, lysogenic cycle is longer. However, there's a big difference between the two. The lytic cycle actually kills the host. The lysogenic cycle does not, because the lysogenic cycle has periods of dormancy or latency where the virus is not active. But the cycles are pretty similar. There's attachment in both of them. So the virus is going to attach to the host cell. Penetration, where they're going to inject their DNA or RNA into the host cell. The lytic cycle has biosynthesis, which is the host cell replicating the virus components. Maturation is when the host cell is basically assembling those viral components into a virus and then release. Now, in the lytic cycle, when the host cell releases the viral cells, it bursts open. Lysis means burst. So lytic cycle, lysis, maybe that will help you remember it. The lysogenic cycle has the same steps except for an added process. So attachment and penetration are the same, but then integration is a new step. Now, integration, basically what happens is the virus still penetrates and injects their DNA or RNA into the host cell. But then what happens is that DNA is integrated into the host cell DNA. Then the steps continue on. Biosynthesis, so now the host is making viral cells. Maturation, they are assembling the viral cells and then releasing the viral cells. So the big difference between the two is that integration. In the lytic cycle, the host cell is just going to synthesize new viral particles, put them together, and blow up and release them. The lysogenic cycle, however, actually integrates the DNA or RNA into the host cell's DNA and then synthesizes, assembles, and releases. Now they are specific. So viruses only attach to their specific host cells, and that's important to remember. Again, penetration, they're going to inject their DNA or RNA. And integration, the virus is not actively being replicated because it is busy integrating its DNA into the host cell's DNA. And then again, the host cells were going to burst open. Now, the lytic cycle is going to basically attack all of the cells in the host and kill it. The lysogenic cycle, again, there's going to be periods where the virus is not active. So not all of the host cells are going to burst and release the virus. Only some of them will, because others are going to be in that dormancy period. So here's a little animation for you guys to see how viruses actually replicate. So there it's attaching. Here it's penetrating and releasing the DNA or RNA into the host cell. There the host cell is maturation and assembling the viral particles and then releasing it by lysis. So once again, the steps of the lytic cycle: attachment, penetration, replication of the viral parts, which is biosynthesis, assembly and maturation, so there's your new little viruses, and then releasing by lysis. So again, it results in the death of the host cell. Virulent viruses, so virulent ones are ones that are really deadly, only go through the lytic cycle. The lysogenic cycle, the first steps are just like the lytic. But again, it does not destroy the host cell, and that's the key. The nucleic acid actually joins the cell's DNA and becomes part of the host cell's DNA. So that's that integration step. Now this can go on for years, and these are going to be your temperate viruses. So HIV, cold sore, shingles, things like that. They are capable of using both the lytic and lysogenic cycles, but the lysogenic cycle is going to be in their best interest because they don't kill the host cell. So they can proliferate for years, as it says. Now, HIV is one of the ones that we're going to talk about when we get to the immune system, because HIV attacks the specific cell in the immune system, and that's why it is so effective. Cold sores are actually herpes simplex type 1, and those recur and then go into dormancy. And just like herpes simplex type 2, if you get that, it never goes away. So genital herpes, you're going to have periods of flare-ups and then remission. And in remission, that's when the virus is actually dormant. Shingles is actually a secondary infection from chickenpox. If you do get chickenpox, if you have the chickenpox virus, I had the chickenpox when I was in 5th grade, which means the virus basically is still within me. It stays dormant in your central nervous system so that your immune system can't really get it. And then if you are really stressed or conditions become right, especially stress, shingles will be a secondary infection. Now, I had shingles when I was 24. It's very painful, and it's basically treated with the same antivirals that herpes is. It's the same family of viruses, so even though they have different courses of action, the same antivirals will work for all of them. So here's both cycles together. You got the lytic and lysogenic cycles. So the lysogenic cycle just has that extra integration phase, as I said. So if the virus is DNA based, you're going to have attachment, penetration, biosynthesis, maturation, and release. However, if it is RNA based, you're going to have attachment and penetration, but then the integration has to include reverse transcription. Remember, transcription is DNA making RNA. So reverse transcription is going to be RNA being turned into DNA, basically. Now, a lot of these viruses are going to have an enzyme called reverse transcriptase, and that's what's going to facilitate the reverse transcription. HIV is one of those. HIV has an enzyme called reverse transcriptase that causes its RNA to be integrated into the host cell's DNA, and now it makes DNA. So that's why HIV is so effective. And then we're going to talk about the other reason in a little bit. And then again, biosynthesis, maturation, and release. So retroviruses like HIV are going to go through reverse transcription. Penetration is what removes the coat, and then the nucleic acid is going to enter the cell. And again, release, it's going to, instead of completely exploding and destroying the host cell, what will happen is it will kind of bud out, like budding in yeast cells. It will bud out and gain an envelope coat from the membrane of the host, and then the viruses will go on and be released. So here's an overview of some different viral infections that you guys can look at. You got the common cold, different eye infections, gastroenteritis, sexually transmitted diseases, hepatitis. Now keep in mind, viruses cannot be treated with antibiotics. Viruses cannot be killed, basically. All you can do is treat the symptoms. So, for example, herpes we were talking about. Whether it is type 1 or type 2, it doesn't matter. You can only treat the symptoms. So if you get a cold sore, you're going to treat the cold sore. If you have a flare-up, you're going to treat the flare-up. But that doesn't get rid of the virus. You will continuously have flare-ups of cold sores or sores in your genital area, depending on the type, for the rest of your life, basically. Now, how often you have flare-ups depends on your immune system and depends on your lifestyle. A lot of factors, especially stress, go into it. So again, antivirals, not antibiotics, are going to be used to treat the symptoms. Now, there are antivirals that treat herpes, influenza, and HIV, but they've actually had the most success with HIV. They use various cocktails of antivirals, so they use multiple medications to treat HIV. Problem with HIV is it mutates and evolves pretty rapidly. Reverse transcriptase, the enzyme that lets them develop, is not very accurate. So oftentimes there are mistakes, and those mistakes basically make new lines of the virus, unfortunately. So how do we prevent these viruses? Well, some of them have vaccines. It can be prepared using a weakened live virus, which is called attenuation, or we can use heat-killed viruses or subunits of the virus. And basically what's happening is you're injected with the virus, but it's not one of the viruses that's going to kill you. It's not virulent. So they've taken away the power of the virus, basically. But they're injecting you with this weakened version of the virus so that your body can develop an immunity to it. Live viruses do lead to better immunity, but they can cause disease, so we have to be careful with those. Some vaccines are in continuous development because of the high mutation rates like influenza and HIV. The problem with HIV, again, is it mutates rapidly. Influenza mutates rapidly. So every year when you get your flu shots, oftentimes it's only against a few strains of the flu. They're the ones that they have predicted are going to be the biggest problem. Public awareness is a big one. Make sure that you are protecting yourself in sexual situations, of course, and make sure that you are washing your hands. A lot of things going on with the coronavirus right now, actually. Social distancing when it gets bad like this, pandemic wide. But when it's just regular things, then just good hygiene, washing your hands, things like that, that's going to help you not get these viruses. And then vector control. Viruses are sometimes carried in vectors like mosquitoes and fleas. So to control those populations is a big thing. One of the big things in the summertime is not to leave standing water. Mosquitoes love standing water. So if you have any standing water in your backyard or anywhere, make sure you get rid of it. Okay, immunity. Now, we have two types of immunity: innate immunity and adaptive immunity. Innate immunity is what we are born with. Innate means like true inborn kind of thing. So it is everything we're born with, so physical barriers and internal defenses that respond to all pathogens. So we have physical barriers like our skin, hair, cilia in our respiratory tract, mucous membranes, mucus and chemical secretions that are going to help get things out of our respiratory tract and other places, digestive enzymes, and stomach acid. The pH of our stomach is about 2, and most pathogens do not like the pH of 2. Then we also have internal defenses like our inflammatory response, complement proteins, phagocytic cells that are going to kill stuff, and natural killer cells, which we'll talk about in a little bit. Adaptive immunity is highly specific, on the other hand. These are the antibodies and the humoral immune response that we have. So we have a cell-mediated immune response, we have antibodies, and then we also have memory, which basically means that if you've come into contact with a pathogen before, our body will remember it and the response will be faster. So here's a chart to kind of help you remember non-specific innate immunity, which is natural, versus specific immunity, which is adaptive and acquired. And then we have the first, second, and third lines of defense. The first line of defense being our skin, mucus, hair, washings. So, when we urinate, when we vomit, tears, things like that. Chemical secretions that we have, and especially in our sweat. Second line of defense are internal things. So complement, inflammation, phagocytes, interferons, things like that, which we'll talk about in a little bit. And then we have the third line of defense, which is our specific immunity. T cells and B cells are specific lymphocytes that are developed. And we have cell-mediated, which are T cells, and humoral, which are B cells and antibodies. And we'll talk about all of this. So the first line of defense is our innate, non-specific, external and chemical barriers, like the skin, mucus, hair, cilia, saliva, tears, and our normal flora. Normal flora is bacteria that we have that naturally live on our bodies. Then we also have the second line of defense, which is our internal defenses. So phagocytes, which are white blood cells, neutrophils, and macrophages, they will come and engulf pathogens, basically. We can release cytokines and interferon, which will destroy the pathogens. They'll poke holes in them, basically, and destroy them. Inflammation, we have localized redness, swelling, heat, and pain. And basically what happens is when your cells get injured, this is how our body reacts with inflammation. And remember the 4 signs of inflammation, redness, swelling, heat, pain. And then complement. Complement is activated by interferon, and it basically complements the innate and adaptive immunity. So it's an extra helper, basically, for what is already going on. Adaptive is also known as acquired. This is much longer than the innate response. And as I said earlier, it's more specific. So we have two types, cell-mediated and humoral. Cell-mediated is called cell-mediated because the cells directly attack the pathogens. It's controlled by what are called activated T cells. There are three types: helper T cells, cytotoxic T cells, and suppressor T cells. Helper T cells do exactly what the name implies. They help the immune response. They facilitate it. Cytotoxic T cells are the T cells that directly go out and kill stuff. Now, HIV, as I was talking about earlier, attacks the helper T cells. And without the helper T cells, you're not going to have an immune response. The helper T cells facilitate the immune response, so you knock them out and you have no response. T cells are lymphocytes that mature in the thymus gland, which is why they're called T cells, thymus T. The humoral response, on the other hand, is controlled by activated B cells and antibodies. B cells are lymphocytes that mature in the bone marrow, hence B cells, bone marrow. But they create antibodies, and then antibodies are going to bind to their specific, what is called antigen, and mark it for destruction, basically. So here's another little chart, hopefully to help you remember. The adaptive immunity, you have two arms, cell-mediated and antibody-mediated or humoral. Cell-mediated is direct physical and chemical attacks. Antibody-mediated, you have circulating antibodies that are going to mark them for destruction. So an antigen is a molecule that stimulates a response from the immune system. B cells produce antibodies when they're stimulated by foreign antigens. So we all have markers on our cells so that we can recognize self cells. And anything that is not a self cell, our immune system is going to attack. So an activated B cell is going to produce an antibody, and that's called a plasma cell. It will also generate what are called memory B cells. Now, memory B cells are going to remain inactive until they encounter the antigen again. So this allows for more rapid response when you are re-exposed to the same antigen. So in a nutshell, what's happening is you're exposed to an antigen, a foreign antigen. B cells are going to produce antibodies against that foreign antigen, and they'll produce memory B cells against that foreign antigen. So when you come into contact with that antigen again, the response is going to be faster and stronger, and it's going to knock it out faster, basically. So an antibody is a protein. It's also called immunoglobulin, It's basically produced by the plasma cells, and it's going to bind to the antigen and mark it for destruction. So think about, like, if you've ever seen a forest and trees have X's on them, those trees are basically marked to be cut down. So that's what antibodies do. They mark the foreign antigens for destruction. We also have what's considered active versus passive immunity. Active immunity is the production of antibodies by plasma cells in response to an antigen. So basically, this is from the individual making antibodies or a vaccination. Passive immunity, the antibodies come from an outside source. So the antibodies are borrowed, that's spelled wrong, and memory cells are not created. Antibodies pass from the mother in the fetus, for example, or if you get a direct injection of antibodies. So adaptive immunity has that memory component, which is going to allow for a faster response when you're exposed to the same antigen. So you have the primary versus the secondary immune response. The primary immune response occurs when you're exposed to that pathogen for the first time. It's going to activate B cells and T cells, but it's going to take time to develop those antibodies and effector cells. Some of those T cells and B cells, however, are going to develop into memory cells. Secondary immune response is when the memory cells become those effector cells immediately upon re-exposure to that same pathogen. Vaccinations are actually used to create memory cells, or it's going to occur when you're exposed to the pathogen. So basically, you're going to be exposed to a pathogen. Your body is going to create memory cells. So when you are exposed to the same pathogen again, those memory cells are going to jump into action, and it saves time because you don't have to create them, and they're going to attack the pathogen immediately. So this is just a chart showing you the primary immune response versus the secondary immune response, the concentration of antibodies. So as you can see in the primary immune response, the antibodies secreted from the plasma cells, and their levels are not very high. However, when you're re-exposed to that pathogen, the memory cells immediately differentiate into those antibody secreting plasma cells. So you have a lot more antibody for a longer period of time. The lymphatic system is also important. The lymphatic system basically runs parallel with the blood vessels. Lymphatic fluid, or lymph, is basically interstitial fluid, but it's located inside of the lymphatic vessels. The lymphatic system as a whole is made up of lymphatic vessels, lymphatic tissue, and bone marrow. It's closely intertwined with the immune system, and we'll talk about why in a second. But the lymphatic tissue are lymph nodes, tonsils, spleen, thymus, Peyer's patches, malts. There's kind of lymphatic tissue all over your body. The point of the lymphatic system is anything that falls out of the blood, the lymphatic system grabs it and dumps it back into the blood, so we don't lose anything that is vital to our survival. So the lymphatic system is going to filter that lymph, and the lymph nodes that we just talked about have a bunch of immune system cells that filter the lymph and help kill any pathogens that may be in that lymph, and then again dump it back into the blood. So the immune system has to be regulated, though. We do not want the immune system attacking self cells. We do not want unnecessary responses to stimuli that don't hurt us. Immune tolerance is the acquired ability to prevent an unnecessary or harmful immune response when you are dealing with self-antigens or things that aren't going to cause a disease, so they're harmless. So once we develop this immune tolerance, our immune system is regulated. However, we do have disruptions in the immune system. The immune system functioning properly is essential to our survival. Immunodeficiencies are failures for the immune system to respond properly. Primary immunodeficiencies, like severe combined immunodeficiency disease, also known as SCID, is a condition which children are born with, and they do not have functioning B cells or T cells. If you do not have functioning B cells or T cells, essentially you do not have an immune system. So everything can get you sick. So you might have heard of the boy in the bubble. The boy in the bubble had SCID, and he had to live in this bubble because he didn't have an immune response. Anything would have made him sick and potentially killed him. Acquired immunodeficiency, on the other hand, is caused by exposure to certain diseases like HIV, or chemical exposure like chemotherapy. Extreme stress can cause it, and malnutrition as well. Now, HIV is one of those that, again, it destroys helper T cells, so it completely knocks out your immune response. Now, HIV itself isn't what kills you. It is the opportunistic infections like pneumonia that you can get because you don't have an immune system response. So pneumonia that you would normally recover from instead will kill you, unfortunately. So that's why they are working tirelessly to try to combat this disease and constantly coming up with different cocktails of drugs to take to try to help keep it in check. And they have it to the point where a lot of people, their levels of the HIV are actually undetectable in their blood. So we've come a long way with the HIV treatment. Finally, we have hypersensitivities. These are maladaptive immune responses towards harmless foreign substances or self-antigens. Now we have three main different types. Immediate hypersensitivity is a response that occurs immediately after exposure. Now, it can be life-threatening. Now, we're talking about allergies. So you might be thinking life-threatening, allergies, I sneeze when I go around pollen or grass. Well, no, we're talking about more life-threatening, like anaphylactic shock, bee stings, insect stings, anything like that, where the toxin is directly injected into your blood. Some people, not a problem. Other people can go into anaphylactic shock. Now, epinephrine is the drug of choice to treat this. My son is allergic to all sorts of insect bites, any kind of insect bite, and he can go into anaphylactic shock. One time he was stung by about 12 bees all in his neck area. His tongue started to swell up and he couldn't breathe, and I had to inject him with the EpiPen. Epinephrine immediately dilates the vessels, so everything opens up and swelling goes down and he could breathe again. Still had to take him to the hospital because you're injecting someone with epinephrine, which speeds up your heart rate, but it solves the immediate threats. Regular allergies you can treat with antihistamines. So if you are allergic to pollen or grass or dust, things like that, Allegra. Now, delayed hypersensitivities take a couple of days after the secondary exposure to occur. So these are like poison ivy and poison oak. If you have dermatitis, some kind of sensitivity to jewelry or cosmetics. They're called delayed because they take a couple of days after the exposure to actually kick in. The last type is autoimmunity. This is sensitivity to self-antigens. You develop autoantibodies and your body basically attacks itself. So the antigen will bind with the antibody, or I should say the antibody will bind with the antigen, and it will form an antibody-antigen complex. And these complexes then will deposit themselves in various areas. So in the case of like lupus, these complexes will deposit in the kidneys and other places. And it's very dangerous, and there really isn't a cure for these because it's your own immune system attacking yourself. With MS, we talked about this with the nervous system. MS is a disorder where your immune system basically attacks the myelin sheath surrounding your neurons, and it destroys transmission. So all of these hypersensitivities can be, at a minimum, irritating, but also deadly. Okay, so that is the immune system. Hopefully the charts will help you keep everything straight. So the biggest thing is with immunity, we have innate immunity, which you're born with, and we have adaptive acquired immunity, which you develop over time. Innate immunity, like our physical defenses, like our skin, hair, mucus, cilia, pH of our stomach, things like that. Complement, remember inflammatory response, got redness, swelling, heat, pain. Those are the 4 cardinal signs. And then adaptive or acquired immunity, you have 2 types. You have cell-mediated and you have humoral. Cell-mediated are the cytotoxic T cells directly attacking those cells. And remember, the helper T cells facilitate this response. So you knock out the helper T cells like HIV does, you knock out the immune response. You have humoral immunity, where plasma cells create antibodies. Now, these antibodies are very specific for their antigen. So an antibody will be created for that antigen. Memory cells are also created, so that secondary exposure, the response is faster. Now, with cytotoxic T cells, they're going to directly attack and kill these pathogens. Now, remember one thing, the antibody attaches to the antigen and basically marks it for destruction, and then the cytotoxic T cells are going to come in and destroy it. So that kind of sums it up. If you have any questions, please send me a message. We will talk more later. Bye.